Hello and welcome my dear students and biochemistry learners. Welcome to this video definition series in biochemistry part 4. In this video we will study some very simple and basic definitions in nucleic acids and nucleotides. So let's begin. Nucleic acids. Nucleic acids, DNA and RNA, are the polymers of nucleotides, polynucleotides, held together by 3' 5' phosphodiester bond or phosphodiester bridge. Nucleotides. A nucleotide is made up of 3 components. First, a nitrogenous base, either a purine or a pyrimidine, a pentose sugar, either ribose or deoxyribose, and third, phosphate groups esterified to the sugar. So nucleotides are nucleoside plus phosphate, simply base plus sugar plus phosphate. Nucleoside. When a base combines with a pentose sugar, a nucleoside is formed. Simply, nucleosides are base plus sugar or B plus S. Nucleotide analogs. Nucleotide analogs are the synthetic analogs of purines, pyrimidines, nucleosides and nucleotides formed after alterations in heterocyclic ring or sugar moiety. These analogs are highly useful in clinical medicine. DNA or deoxyribonucleic acid. DNA is a polymer of deoxyribonucleotides. It is composed of 4 deoxyribonucleotides. They are deoxyadenylate, deoxyguanylate, deoxycytidylate and deoxythymidylate. So nitrogen bases A, G, C and T are present. Uracil U is absent in DNA. Denaturation of DNA. The 2 strands of DNA helix are held together by hydrogen bonds. Disruption of hydrogen bonds by change in pH or increase in temperature results in the separation of polynucleotide strands. This phenomenon is known as denaturation. Melting temperature of DNA. Melting temperature or TM is defined as the temperature at which half of the helical structure of DNA is denatured. So 50% denaturation. At lower temperature, the melting strands are reassociated. This is called annealing. Both denaturation and annealing are the terms which are of significance in PCR, polymerase chain reaction. Nucleosomes. The DNA double helix, approximately with 150 base pair, is wrapped around a histone octamer formed by H2A, H2B, H3 and H4. This super twisted helix forms a spherical particle of 10 nanometer diameter called nucleosome. Chromosomes. A group of nucleosomes form the DNA fibrils. About 6 such fibrils are further supercoiled to form 30 nanometer diameter chromatin fibers. These fibers are further supercoiled and condensed to form chromosome, obtained from the Greek word chroma for color, soma for body, chromosomes, colored body. and this happens during M phase of the cell cycle. So chromosomes are visible and formed during the M phase of cell cycle. In humans, there are 23 pairs of chromosomes. Gene. A gene is the entire DNA sequence required for the synthesis of a functional protein or RNA molecule. In addition to the coding regions, exons, a gene includes transcription control regions and sometimes introns. So transcription control regions are the promoter region, operator regions and all. Exons and introns. These segments of the gene coding for proteins are called exons, expressed regions. The exons are interspaced in the DNA with stretches of silent areas called introns, intervening areas. So exons get expressed and introns are the silent regions, they do not get expressed. Cistron. A cistron is the unit of genetic expression. It is the biochemical counterpart of a gene of classical genetics. One cistron will code for one polypeptide chain under the concept of one cistron, one polypeptide. RNA, ribonucleic acid. RNA is a polymer of ribonucleotides held together by 3' to 5' phosphodiester bridges. It has ribose sugar and nitrogenous bases adenine, guanine, cytosine and uracil. So nitrogen base T, thymine, is absent in RNA. Ribozymes. In certain instances, the RNA component of a ribonucleoprotein, that is RNA in association with protein, is catalytically active, and such RNAs are termed as ribozymes, RNA enzymes, so ribozymes. That's all, friends, for today's video. I am thankful to all these resources for making this video possible. Thank you, friends. Thank you for watching this video. Be happy, healthy, stay safe, and remember, biochemistry is easy. Thank you so much.